Okay, welcome everybody to Open Source Antibiotics Series 2 meeting on Friday, October 2nd. So thanks for everyone for coming along. Here is the issue on GitHub that talks about all the things that are going on in this series at the moment with the various action items. So I think the main thing will just be update on chemistry and some compound design things, but just as an update, we got this update from Lee Graves at UNC about the mechanism of action. So the compounds that we sent over, one active and one inactive, we previously talked about the need to make sure that the compounds were active and inactive in their assay, because I think they're using compounds that have been in the freezer for a while or fridge for a while. So they had them tested in a kind of Mercer assay that's being run over there, and they were fine. So the active was active and the inactive was inactive, kind of. The active was 10 micromolar, which I think is not actually that far off hours when you do the calculation. It's a different unit to to ours, which is a micrograms per mil or whatever it was. But I think it's an active compound, so I think it's fine, and it's a good control for the mechanism of action study. So those experiments are starting now, and I reckon we'll have data at the end of October, which is really good. So they're underway, which is fantastic. For the in vitro stuff that we talked about last time, we still have this thing which one of us is going to have to solve about the compatibility of the units, which I haven't looked at, just to make sure that we're talking about the same things. But this second point was the crucial thing from a crucial thing from last time, was that the compounds that we were interested in for the metabolic ID studies and more investigation of clearance, we had thought we were going to use the main active, which had the benzothiophene in it. But it turns out that that compound is just cleared ridiculously fast and actually isn't that interesting. And we should go for a compound which has, fast but not too fast clearance, which is more typical, I think, of the series, which lacks the sulfur. And so I think we've become much more interested in this benzofuran compound, which I think was number compound with the 821 at the end. So I guess we need to check that that compound was available. That's right, isn't it? That we're going to have plenty of that compound to send out to people if they want to study it.
I think we'll have to remake it, right, Ed?
Yeah, we'll have to remake it. I just finished the boronic ester of it, so you can do the Suzuki.
Great. Okay, fantastic. Yeah, I think it's going to be a good one to go for, I reckon. Just because it's more representative, and it'll have metabolites that are more typical. All right, good. For the screening, we've lined up a bunch of another assay screen for potency for October. I think maybe in a week's time or so was the last thing that Paul said. And I'm in the process right now, as of yesterday evening, finalizing our ability to do some tox screening in a lab in the School of Pharmacy. So that's progressing, and we're nearly there. Biofunctionalization, that was held up because of the results from last week. Because I think we need to send compound 821, so that paperwork will go in today, and we'll finalize that. And I've pinged this week GSK to check that I can paste a bunch of stuff about some history of this series. I'm still waiting to hear back, but I'm pursuing that still. We need some kind of video from you guys if you're willing to put on your best outfits and go on camera to say something. So maybe we can try and pursue that this week, just a little kind of video of something that's going on. Someone in the lab's buying a webcam, right, for problem session. Maybe we could use that or something. I don't know. But just something that we can put up there, simply. I've contacted Bill about the original code report, waiting to hear back, but I sent an email this week about that. And the compound updates, the compound donations, they've arrived, right? So Dana, sorry, is that all of them have come in, or are we still waiting?
No, the ones from Imperial I don't have, but the other two, they've come in.
Right. Okay. And the Imperial ones, is that just because we're waiting, or do we need to check?
I'll send an email because I don't think we've gotten confirmation that they've shipped them yet.
Okay.
So I'll follow up on that.
Right. Okay. All right. Good. Thanks. And then I guess we can just transition quickly into synthetic chemistry if you guys are happy to update.
Mhm.
All right.
Okay, so I'm still trying to make the boronate of the imidazole. So I did go back and do some close slit examinations. There's a lot of tin couplings at this 4 position, which is how people are getting heterocycles on there, which I kind of wanted to avoid, but I did find an example where they did the borylation in DMF instead of dioxane. So I tried that. And these reactions too are a little bit hard to monitor because they don't they don't ionize very well and they're not all that UV active. But I took a crude NMR. It looks like the starting material is gone and there could be product. So I took that crude and I didn't try to purify it. I just pushed it to the Suzuki. So that I just need to work up today and then try and figure out whether this reaction has done anything.
Right.
Then in parallel to that, I was also looking at with the trityl group on because there are several examples of that. So this reaction I put in the microwave, it does look like it went a little bit better than conventional heating. So I think I'm going to try the same thing with the crude material here. Because it was similar, it does look like the starting material has been consumed and it could have some product. Then this at the bottom I just finished up from last week. This compound, which is sort of a bridge between the DNDi compounds and then the 822, the active with the benzothiophene on it.
Uh-huh. Is that what that? So 144. Wait, we don't have that. We haven't had that compound before. Is that right?
Yeah, right.
Huh.
So
Wow.
Alvaro made that with the matching SB, so the whatever methylene dioxy, that group instead of the benzothiophene.
Oh right, sorry, with the 2 methoxy.
Yeah. So it's this core, but matched back to the SB compound instead of the
yeah, right.
active.
Okay. So
yeah.
this is the double check, right?
Mhm.
Okay, great. Okay, and that went okay.
Yes. So I had to purify it a couple times, but, and I'm just waiting on the NMR, but I think it's fine.
Okay, awesome.
And then the borylation. So looking up going forward, if these turn out that they haven't worked, I did find this isopropyl magnesium chloride prep of the boronic acid. So we have the trimethyl borane, but not the Grignard. So I ordered that. So that we can try. And then if none of that works, I can try this sort of pathway B, which just puts in the 2-pyridyl in sort of this one pot procedure. But that we have to order both of the starting materials. So that's the plan.
And that's a wait, is that KCN as a reagent there in the bottom?
Mm-hmm.
Oh, yeah.
I think it's catalytic. I'll have to go back and check the prep. It might be less than 1 equiv.
Okay. Wow.
Yeah. So I was trying to avoid that one.
That's why it's B.
Mm.
Okay. All right. Great.
And then, so for my stuff, so these 2 compounds up here are done, both in 41%. So they're good. So I tried to do this acyl bromination this week, but I kind of gave up on it because it's just really messy. Not very nice to work with. But there's another prep which uses this pyridinium perbromide, which is a lot easier to handle. And I think based on the patent, the reactions also a lot easier. So I've ordered that. So I'll try that when that comes in. And then that'll make, I guess with this core on that one. I've ordered some of this Bach protected indole because I guess we're shifting to the to this one. So I mean, we might as well have that for another comparison. So that's coming in.
We don't have that already.
No.
Oh wow, gosh, that's I didn't spot that.
Yeah, and the oxygen and the sulfur are both active, so I think we might as well try the nitrogen too.
Huh, great. Okay, great.
Yeah. And then for these cores, so I've made all of these. So I just need to do Suzuki's. As I said before, so I made the boronate, the benzofuran. There's a bit of the B2-pin2 left in it, but it's more trouble to try and purify it, I think, because it just co-elutes. And then, yeah, so these are the 4 target compounds from that. Do you think we still need to make the benzothiophene versions of these? Since I guess we don't.
Is the
really
benzofuran active?
I would guess not. I mean, it's so undesirable. No?
Well, we could pop we could pop the if you have that paramethylthio compound was pretty potent. The one the metabolites are made of, so you could just use that as the top side.
I mean, if we get potency with these, then
because do we know if benzofuran? Because you really want to see if the ring substitution of the middle piece kills potency, right? So do we know if the benzofurans are potent?
Yeah, well, there was the one right from last time.
Yeah, so this one.
Yeah.
So that is potent.
Yeah, not as potent as the benzothiophene, but it's not bad.
Ah, I was gonna say just use the one they made metabolites of. Just the paramethylthio phenylboronic acid, but I'm not sure.
Yeah, I mean, I don't know. Feels to me like if we get potency from some of these, then we're in the clear.
Okay, well, I guess, yeah, I'll make these 4 first and then see what happens.
I mean, if they're all bad, then we think, but hope for the best. Yeah.
Yeah, hopefully the control compound actually shows some activity.
Yeah. Great.
Can I just ask a quick synthesis question?
Mm-hmm.
Have you tried doing a Nagishi coupling here instead of Suzuki?
No.
Because it seems that you're taking aryl bromides and forming pinnacle ester, the boronate esters.
Yeah.
And then coupling. And yeah, Suzuki's are at the coupling stage, the Suzuki's are maybe a little bit more robust than the Nagishi coupling, but if the Nagishi coupling works, it's one step. Yeah.
Yeah,
so
we can have a look into that.
Just thinking, and I was going to have the same comment on the previous slide. Yeah, where maybe if you go back to 17. Where was it? Yeah, you try if you're having trouble making the boronic ester of the imidazole.
Yeah, I think that's
making that compound 1451. You know, you've got the 2 bromides, pyridine, and
Yep.
So although that free NH on the imidazole could be causing you issues, so you might want to protect it. I'm trying to think what SM groups can sort of protect. Might be able to protect the imidazole.
If you do an Nagishi coupling, though, how stable is that 3-pyridyl zinc compound then?
Well, that's the trick. Is that the reason that they're not as ubiquitous as Suzuki is because those zinc intermediates are unstable. But it is a one-pot. But it does turn into a one-pot reaction.
I don't remember noting down any examples of Nagishi couplings with this motif, but can do a more targeted lit search then.
I mean, you would do what you've done here is what you would do first, definitely. But if it's not working,
yeah, it's yeah, that's a good idea. Thanks, Ben.
thanks. Okay, and then just I think there's that is pretty much it for today. But let me just double check. Yeah, because that's all the things we were talking about there. And then we're still waiting on a reply from Paul about the comparison of methods that were used between his lab and COAB, and also what is the likelihood that the in vitro assay would yield some oxidized metabolite, which seems like a low possibility, but we wanted to make sure that we weren't missing something about the assay. And I think that's everything. So, I mean, I think what we're doing at the moment is planning for, another screen in about a week's time or so, and seeing if we can build the potency up, because that will inform what we then send for next in vitro PK. And we're awaiting the MOA stuff. So I guess we're sort of taking care of everything, I think. Did anybody have anything else here that we should be talking about? I think that's everything.
I just had one other synthetic suggestion.
Yeah.
Sorry, guys.
No, that's good.
I'm going to need to see the structures again. Sorry. I was just wondering if you thought or looked up in the literature whether it's possible to form the 2,3-aryl substituted targets. So let's look. Okay, compound 1441, for example, right? The one at the bottom. You can imagine forming that in a 3+2 cycle addition from the alkyne and aminopyridine. Right? So if you were to disconnect,
I think Dana posted that.
Possibly, that's why I mean, I haven't been following the work you've been doing, but I look at that, and for me, that's the obvious disconnection.
So I think I was thinking a little bit differently, because I was going to try and do a Sonegashira. No, sorry. What am I trying to?
Yeah, you'd use a Sonegashira to make that reagent.
Oh, sorry. So between the thiophene and the pyridine is what I had thought of, and then making triazoles as sort of an alternative core. But you're talking about something a little bit different. I'm talking
about disconnecting. So if you can look at 1441 and basically cut it so that you've got a 2-aminopyridine, and then a triple bond between the pyridyl ring and the thiophene.
Okay.
Yeah, you make that, and then and is it known to make those compounds, using a 3+2? Now you've got the regiochemistry problem, right, which is you're probably going to end up with a mixture of the pyridyl and the benzothiophene at the two positions, and then how using NMR, you're going to have to use NOE to be able to distinguish between the two and hope that it works. But I'm just wondering if it's worth doing a literature search to see if that's known.
Yeah, I think, yeah.
I'm just throwing out there as an idea, but the kinds of coupling you're doing here, the Suzuki couplings from that 3-brominated position, those work, right? So yeah, I'm just putting them up. I'm just saying if you're having trouble with making some of these targets, that might be the way to do it. And also the other thing, it was a question, it wasn't clear to me why you're trying to make 145 and 1352, like the compound on the top right. Is that just to see what, I mean, it seems like a fragment, right? So what's your plan with that afterwards?
To brominate it and then put the benzothiophene on. But the question was whether those methylenes of the original core were getting metabolized. So I was just wondering if we could just get rid of them and essentially just use the unsubstituted imidazole as the core instead.
Oh, so going from a 6,5 system core to just the imidazole as the core.
Yeah, yes.
It was flagged as a potential hotspot, wasn't it?
Yes. Yeah, that came up on the smart SIP, and I think a little bit on the metasite analysis also, those carbons.
Yeah, I mean, I would do that. Yeah, it's an interesting approach, but you're cutting off quite a large part of your molecule, and the risk is that you're not. I mean, we don't know how these things are working, but I would. Have you thought about bulletproofing the ring instead and incorporating an oxygen in, or substituting the ring in such a way that you're basically making it a little bit more polar? That would be one way. And then from our, I think I might have mentioned this last time. In our experience, when we had real bad clearance issues with these sorts of compounds, the way we got rid of it was by turning the pyridine into something else.
Right.
So it's not even if I mean, yeah, I would take metasite results with a pinch of salt.
Okay.
Let's put it that way. Yeah.
Yeah, I think we were thinking that the starting materials for those the types of compounds you were talking about would be a little bit hard to come by, but if we switch to the one pot protocol, that probably would be easier to make modifications there. So yeah, maybe that's something that we should think about in future.
Regarding the triazole, I noticed that your Suzuki failed. I did do a literature search, and there were some other conditions I think I probably posted in the GitHub. Also, I guess if you do make that alkyne, you're just gonna react to a TMS azole. I got a TMS azide and then hydrolyze off the TMS to get the unsubstituted triazole.
Mm-hmm.
I don't know. I thought it was an interesting idea, but I'm not sure if it's going to give you hell in the lab, you know?
Yeah, that's kind of my feeling about it too.
You know, I always could try switching out the palladium from DPPF to something else, but at this point, it seems it's much like the molecular bio literature where everyone has their own secret sauce.
The compound donations are playing a lot on the 144 structure, right? That's the majority. So we're going to get a lot from that, I think, in sense of clues as to what might be tolerated, which is going to help, I think. You know, and some of the judgments we made early on about the SAR were a little bit skewed because we assumed that a lot of these things were active, and actually maybe, sorry, assumed a lot of these things were inactive when actually they may have been active.
Yeah, and the benzodioxole might have killed potency.
Yeah, so at this point, we you know, it's about acquiring more SAR data. I think that's the most important thing, which we
because we're basically at almost square zero now.
Well, not quite, because you know because the benzofuran's active, so we've got something that we can stick there, but we do have to take all the negative data.
Yeah,
inherited with a pinch of salt.
I think we can say a lot about the sort of benzothiophene, benzofuran, basically with the blue, everything that's blue on GitHub. That we can say a lot about because we can compare back to, you know, 822. But the core and then the pyridyl stuff, that is a little bit more of a question mark.
Yeah.
If you have just toluene boronic acid, it might not be a bad idea to change the top piece of that from benzofuran to that, because we're not or do we know that switching that benzofuran to alkylphenol will kill potency?
So I've made just the phenyl ring instead of the benzothiophene. That hasn't been tested yet.
Ah, okay. I was gonna say it's para CF3, the usual.
Yeah. So we don't
interesting things to come then.
That's just this. So yeah, we've got the methoxy up here. I'm not sure I can expand the screen. Sorry, I think it's.
I see, but it's interesting though how the paramethoxy compared to the parathiamethoxy, the potency goes up so much between the thiomethoxy and the methoxy, para substituted. If you just put a para CF3 in there, or maybe even a CF3O, that would be very interesting, I think.
Yeah, this is our 821 compound that is.
Yeah, I'm talking, I was saying the 823 and 824 compounds. If you just put a CF3 there at the para position, or maybe even a CF3O, if you can find the boronic acid. I can look on Fluorochem.
Yeah, that would be great, actually. I mean, our old friend, the para OCHF2, you know, that was used so much in the
S4 malaria. Exactly. It's exactly.
Okay. Yeah. Any searches would be great.
So I'll do a dump of that later today, I guess.
Great. Okay. All right, good. Thanks, everybody. Really useful. And I'll post some comments to the issue and then post the video and any other things that crop up after we've left the call, just stick them on the issue. Nice to see you all. Thanks very much.
Bye.
See you again soon.
Thanks. Bye.
Bye.